everyone. I am Dr. Manoj Kumar Sharma, Associate Professor, IITS College of Pharmacy. This is lecture number 26 and it is the third part of the designing and methodology topic, and the subject of this topic is biostatistical and research methodology. The code is BP801T and the course is Bachelor in Pharmacy, and the university is Dr. APJ Abdul Kalam Technical University, Lucknow. So, the contents of this lecture are experimental studies, designing clinical trials and various phases of trials. So, these three topics we will read in this lecture. So, first is experimental studies. Experimental studies are one where researcher introduce an intervention and study the effects. Experimental studies are one where researcher introduce an intervention and study the effects. Experimental studies are usually randomized, meaning the subjects are grouped by chance. Randomized control trial or RCT, eligible people are randomly assigned to one or two or more groups. So, in experimental studies, we assign the subjects or patient in two or three groups randomly. Okay. In experiment, the well designed experimental studies can establish a cause and differ effect relationship between the response and explanatory variable. So, in experimental study, we find a relationship between the cause and effect of response and explanatory variable, like smoking cause cancer. And however, using experimental studies is not always possible. In experimental studies, the researcher creates changes in the explanatory variable and note the changes in the response variable, that is, experimental studies are used to answer interventional questions. As we find here, the cause and effect relationship between response and explanatory variable. So, it is main is because we answer the interventional research questions. The definition of experiment, before read to experimental study, we some note to some terms, but first is experiment. In an experiment study or an experiment, the researcher intervene to control the values of the explanatory variable C that are applied to the individual. The researcher allocate treatments, that is, apply the intervention. The researcher allocate treatments, that is, applied to the intervention. This is the figure an experimental study, where we have explanatory variable and it is we find a relation with response variable and we allocate the treatments and people according to our study. The definition treatment. So, treatment are the condition of interest that those in the study can be exposed. Treatments are the condition of interest that those in the study can be exposed or in the comparison or connection. In experimental, treatments are imposed by researchers. तो ट्रीटमेंट क्या है? ट्रीटमेंट एक कंडीशन है जिसे हम अपनी स्टडी में एक्सपोज्ड कर इन ट्रीटमेंट क्या हुआ? ट्रीटमेंट आर कंडीशन ऑफ इंटरेस्ट or that those in the study can be exposed to in the comparison or connection. The experimental study, this is the figure. Now, there are two types of main experimental studies, true experiments and quasi experiment. True, or you can say true experimental study or quasi experimental study. This is, we have a table, in this we comparing experimental design it can have descriptive studies and do not have any comparison or connection groups. So, there are three types of study type, either true experiment, second quasi experiment, and third is observational. In true experiment, researcher allocate who or what to group. That means, which treatment is applied to which person. That means, he allocate what to group, किसको प्लेसिबो देना है, किसको ट्रीटमेंट देना है, ये रिसर्चर डिसाइड करता है. एंड आल्सो रिसर्चर एलोकेट ट्रीटमेंट्स टू ग्रुप, और ट्रीटमेंट ग्रुप्स में किनको रखना है, किनको नहीं रखना है, किनको प्लेसिबो में रखना है और किनको ट्रीटमेंट में रखना है, ये भी वो रिसर्चर डिसाइड करता है. वाइल इन क्वासी एक्सपेरिमेंट researcher allocate temperature to groups. Be remember, in quasi experiment researcher allocate treatments to group, but not allocate who or what to groups. And in observational study researcher not allocate who or what to group and not allocate treatment to groups. So, this is the main three difference. So, in true experimental study, the true experiment are commonly used, but cannot always be conducted. An example of true experiment is a randomized controlled trial often used in drug trials. The definition of true experiment: in a true experiment, the researcher allocate treatments to group of individual, that is, he decide the value of the comparison or connection used on the individual and determine who or what individual are in those group. That means, first he decide people into treatment and another group. So, first he allocate the person, then while these may not actually happen explicitly, they can happen conceptually. The true if we take an example of true experiment, this is the AKtU study. Barrett et al. Could be designed in a true experiment. The researcher would allocate individuals to one of two groups and then decide which group took an AKtU and which group of did not AKtU. So, AKtU actually a purple color cone, or it is called purple color cone flower, and it is worldwide found herbs, and it is a remedy for common cold, flu or pain treatment like migraine and other health issue. So, AKtU is a very popular herb which is found in worldwide and used for common cold, flu and other health issues. So, in this example, Barrett do a study, a true experiment, and he allocate the individuals to one of two groups, first in placebo or who did not take the AKtU, and second who treated with AKtU. So, this is the true experiment figure, in which first the investigator Barrett divide the people into two groups, group 1 and group 2. First he divide the people into two group 1 and 2. After that he decide the group 1 allocate to use AKtU and group 2 allocate not to use AKtU, and then compare. So, this is the experimental study. Another is quasi experimental study are similar to true experiment, but treatment are allocated to groups that already exist. That is a difference between true experiment and quasi experiment. In true experiment, first investigator divide the people into two groups, then he decide which one to send treatment group and which one send to control group, and after that he decide which group to send treatment or which not treatment. But in quasi experimental study we have already two groups and investigator decide for which group to give treatment and for which is not control or not give any treatment. So, definition: in quasi experiment the researcher allocate treatments to groups of individuals, that is decide the values of the comparison connection used on the individuals, but do not determine who or what individuals are in those groups. We take example, the AKtU study based Barrett et al. 2000 could be designed as a quasi experiment. The researcher would need to find, not create, two existing groups of people, say from the two different suburbs, then decide which group took AKtU and which group did not take AKtU. So, in this you see there we have already two decided group, we have no people to send in group 1 and group 2, we have already existing two group and investigator assign group 1 to use AKtU and group 2 not to use AKtU and then compare the results. So, this is the experimental quasi experimental studies. Third is clinical trial design. The clinical study design is the formation of experiment trials and studies, observational studies in medical, clinical and other types of research involving human beings. We remember clinical trial study design is the formulation of experiment, trials and study, for example, observational studies in medical research, in clinical and other type of research in which human beings are involving. So, clinical trial generally do on human subjects. The goal of any clinical study is to assess the safety, efficacy and or the mechanism of action of an investigational medicinal product IMP or produce or procedure or new drug or device that is in development, but potentially not yet approved by the health authority, for example, FDA, TGA, EMA, PMDA, etc. Clinical trial design: choosing the right design is essential for any clinical trial, and before choosing a study design one must establish the aim and objective of the study and select an appropriate target population that it is more representative of the real world population. These avoid unnecessary expenses, time, delays and improves the chances of approval. When we in clinical trial, there are several types of clinical design used in clinical trial, illustrated below. First is treatment studies, in which we have randomized control trial, blind trial, non-blind trial, adaptive clinical trial and non-randomized trial like quasi experiment. So, there are different types of treatment studies. Second is observational studies. In clinical trial, in observational study we have descriptive study, in which we only describe about the characteristic of the population, for example, case report, case series, population study. So, in observational study you have not do any treatment, but you observe only. In analytical you have do cohort study or prospective cohort study, retrospective cohort study or time series study. Also we do in analytical study is case control study, in which we do nested case control study or cross sectional study. Cross sectional study, the study in which you observe the patient at a point of time. These are generally survey study or community survey. So, when you do a community survey, so this is a cross sectional study. In cross sectional study, you observe the present condition of the patient or the subject. Another in clinical trial observation study is ecological study. There are various phases of in clinical trial. In clinical drug development often consists of 5 phases. When we do different types of clinical trials, we have each clinical trial have different phases. For example, we use clinical drug development, it consists 5 phases, phases 0 to 4. So, we numbering 5 phases from 0 to phase 4. Clinical trial happen in these phases to answer different questions, and each phase builds on the results of previous phases. So, in clinical trial for drug development, we have 5 phases, 0 to 5 phases, and when numbering are it 0 to 4. First phase is phase 0 of clinical trial or phase 0 trial. Phase 0 is designation for optional explanatory trial to conduct in accordance with the United States Food and Drug Administration US FDA. The phase 0 is a designation for optional exposure trial to conduct in according to US FDA. In phase 0, studies generally use only a few small dose of new drug in a few people. These studies give no data on safety or efficacy, as the data is too low to cause any therapeutic effect. So, in phase trial 0 trial, the studies give no data on the safety or efficacy, as the dose is too low to cause any therapeutic effect. Researchers uses phase 0 trials to determine the drug pharmacokinetics. The researchers use phase 0 trial to determine the drug pharmacokinetics and pharmacologically significant dosage of the drug. Also, researchers do phase 0 to understand the drug mechanism of action, and also they help in making go and no go decision on the relevant human models of relying completely on normal data. So, this is the next main aim of the phase 0 trial. Now, phase 1 trial. Phase 1 trial, the primary aim of phase 1 clinical trials are to determine the safety, to tolerability, pharmacokinetics, pharmacodynamics of a compound and early measurement of drug activity. So, phase trial, the primary aim of phase 1 trial, or you can say primary aim of phase 1 clinical trial are to determine the safety, tolerability, pharmacokinetics, pharmacodynamics of a compound and early measurement of drug activity. Now, phase 2 trial. The phase 2 trial usually start with initiation of studies in which the primary objective is to explore the therapeutic effects in patient. An important goal for this phase is to determine the dose and regime for phase 3 trial. So, phase 2 trial is usually start with the initiation of studies in which the primary objective is to explore therapeutic efficacy in patient, and the important goal for this phase is to determine the dose and regime for the phase 3. So, this is the next the use of second trial for third phase. Phase second trials studies sometimes consist of phase second A and phase second B. That means, in some studies phase second divided into two class, phase second A and phase second B. However, there is no formal definition for these two subcategories, but in simple words we can say phase second A study are usually pilot studies. That means, we sometimes to complete our research we do a pilot study to find that our objective are is clear or not, or to verify our objective or to verify the design. So, we do a pilot study in some cases. So, कई बार हम जब अपनी स्टडी करते हैं तो उस टाइम ये चेक करते हैं कि जो हमारे ऑब्जेक्टिव्स हैं स्मॉल पर्सन पे स्टडी को कंप्लीट करके कि वो क्लियर हो रहे हैं या नहीं, अदरवाइज वी चेंज आवर डिज़ाइन. सो दिस इज़ पायलट स्टडी. तो फेज सेकेंड स्टडी आर यूजुअली पायलट स्टडी designed to demonstrate clinical efficacy or biological activity, and it is also known as proof of concept studies. And the phase second B study determine the optional dose of which the drug shows biological activity with minimum side effects. That means, definite dose finding studies. So, in phase second we determine the optimal dose at which the drug shows biological activity with minimum side effect. We want to find maximum output with minimum side effect. Now, phase 3 trials are usually considered to begin with the initiation of studies in which the primary objective is to demonstrate or confirm the therapeutic benefit of treatment or drug or device and etc. The phase 3 is usually considered to begin with the initiation of studies in which the primary objectives is to demonstrate or confirm the therapeutic benefit of a treatment or drug or device etc. Phase 3 trials suppose usually in phase 3 are designed to confirm the preliminary evidence accumulating in phase second that a drug is safe and efficacy for use in the intended indication and recipient population. The studies intend to provide an adequate basis for obtaining regulatory and marketing approval. Studies in phase 3 may also further explore the dose response relationship or explore in the drugs used in wide populations in different stages of the disease or combined with another drug. Phase 4 trial. The phase 4 of clinical trial are conducted generally after the drug approval. Therapeutic use studies go beyond the prior demonstration of the drug, its safety, efficacy and dose definition. These studies are also called pharmacovigilance studies. The studies are not considered necessary to approval, but are often important for optimizing the drug use. Commonly conducted studies include and additional drug-drug interaction also find, and studies designed to support use under the approval indication, for example, mortality, morbidity studies, epidemiological studies. So, in the phase 4 we do all these things. Thank you.